I first want to say to you guys, I guess we're about near the middle of the course, believe it or not, when you get to this point anyway. And I just want to say that this class has been the best online class that I have ever taught. So if you are feeling overwhelmed or if you're worried about how you're doing, overall, this class, you guys are doing a tremendous job on, especially, I'm just so impressed with the e-science labs. Um, you are completing them and turning them in and doing the pictures, and I'm really enjoying grading them, honestly. Um, what I want to show you right now is this is extremely important because some of you are losing like maybe 30 points per lab because you're not doing this. I know that you have a copy, you have like a paper copy of the lab manual. I know you have that with you, but that paper copy doesn't have the lab report in it. It doesn't have the pre and post lab questions. So I want to show you for chapter 10 and chapter 11 on mitosis and meiosis. I'm just going to go through and show you what you have to do for these chapters. Um, in chapter 10 on mitosis, here are your slides and you've got your quiz posted. Um, I haven't done the recorded lectures yet. I'll get on that as soon as I can. But the lab that you're doing is lab 11 on mitosis. This is the assignment that you're going to upload your workbook to, but you have to click on this link. This link right here. You must click on it. You must go to this link. And once you do, you're going to find that there are 3 experiments. When I go down to assignments and procedures, there are 3 experiments for you to do. And the very first thing you're going to do is you're going to click on the lab workbook, download it, and open it up. Because I think this is it. Okay, because this is what I want you to fill in. This is what I want you to fill in and save and then turn in to me. This entire lab workbook or lab report, whatever you want to call it, includes all 3 experiments. So it's really one document that you have to fill out. Okay, so in experiment 1, you're going to click on that. This is something you do online. It tells you in this experiment, you'll look at different stages of mitosis in onion cells. And then you can read through the rest of the lab. I'm not going to tell you how to do it. You can follow the directions. But you're going to click here to view the slide. So you're just going to view these different slides and answer, follow the instructions in the lab. And experiment 2 is when you're going to use your beads. So for experiment 2, you're going to use, you're going to make 2 sets of chromosomes. Let me move my face out of the way. You're going to make 2 sets of chromosomes like this. Now, you don't have to do blue and black. You can do any 2 different colors of the smaller beads. And then the larger beads, you also can do any 2 different colors, but you're going to make a total of 4 sets of sister chromatids. What you're going to set up is going to look just like these 2 pictures. So you're going to have one pair of sister chromatids here, another pair here. You're going to end up with 4 total chromosomes or pairs of sister chromatids. What you need to have on your table or your working surface is this setup right here. So, and then you're going to go through and you're going to simulate mitosis and what happens with the chromosomes during mitosis. And as far as filling in the lab workbook, the part that you have to do for experiment 2 says that you can draw. So in prophase, it says for you to draw what the beads look like in prophase. Well, you can actually take a photograph. In fact, this is what I prefer that you do. Take a photograph of your beads that you have set up in prophase and put that picture right here. And then upload your metaphase and your anaphase and your telophase. You don't have to upload cytokinesis. You can draw that if you want to, or just leave it blank because the chromosomes look the same in telophase as what they do in cytokinesis. So, but instead of making drawings, you can actually, and I would like for you to take pictures. This is how you're going to document that you did the bead activity. Then, so we're going to go back to, and then there's experiment 3, the importance of cell cycle division. And this is something that you do online. So there's nothing, this experiment 2 is the only one that you actually need materials for, and it's just the poppet beads. Okay. Then if you go into chapter 12, you might have noticed on the syllabus that lab 11 mitosis and lab 12 meiosis are due on the same exact date, which is June 26th. When you click on lab 12 meiosis, go to assignments and procedures. You're going to download your workbook. Now this time, you do not have to do the poppet beads. You can just answer the questions and make drawings. You may be running short on time, and you may be a person that doesn't, the beads are not really helping you, they're just making it more complicated. But you will do both experiments and make your drawings this time instead of doing photographs because it's just too many. If you notice, when I go, if I show you all of these drawings that you have to make, you'll see. So there's trial 1 and trial 2, and you have to do all the stages of meiosis. And meiosis contains double the stages that mitosis does. So really, if you just want to do the drawings and forget the beads, that's fine for the meiosis activity. And that's it. Just make sure you are downloading the lab workbook, and that is what you're completing and uploading to the assignments. Because, like I say, many of you are missing up to 30 points per lab because you're not answering the questions in the lab workbook. You're just doing the lab, filling in the table, and that's it. You're not answering any of the pre and post lab questions. So I just wanted to make sure that you were aware.